this video, we are going to talk about food tests. Now, what are the food tests that are performed for the biological molecules we have discussed in the previous video? If you want to know about the biological molecules and their role, do watch the previous video. The link will be shared in the I button. Okay, so basically, biological molecules, there are carbohydrates, then we have proteins, then we have fats, and then we have lipids. So basically, in order to find out that whether the carbohydrate is present or not, we perform Benedict test. So let's find out how we perform these tests. You can see here there is a test tube, and then the test tube has been taken. The three test tubes that I'm going to draw at the bottom, they are representing that there can be three type of results, and the fourth one is going to be the strongest result for the strongest amount of glucose present. Okay, so let's start with the color differentiation first. In the examination, they ask you to either compare different types of milk or different types of solutions in which whether glucose is present or not, or whether glucose is present in less amount or more amount. The first thing that we need to know is the blue color of Benedict solution, it has to change into some other color. Okay, and for Benedict solution or Benedict test, the first precaution that you have to take is that you have to heat the Benedict solution along with your solution in a water bath, and the temperature needs to be set at 70 to 80 degrees centigrade. Okay, this is the first precaution and the safety measure we are going to take. Now we need to know that there are three types of results that are showing different types of different amount of glucose present. If the glucose amount is present in the traces, we will say that green coloration will be formed, which indicates that yes, glucose is present but in a very lower amount. If yellow color will be appeared, it means that there are again moderate amount of glucose present, and this moderate amount of glucose can also be called as a reducing sugar present. Okay, this moderate amount of glucose can also be represented by orange color. Now after these three color differentiation, if we want to see the highest concentration of glucose, there will be the brick red color formation, and that brick red color formation will prove that the glucose solution is present in its maximum amount. Okay, so for Benedict solution, we have to heat the solution. Now we are going to perform another test for carbohydrates. We will be performing it on bread and potato, and it is called as starch test. What happens in starch test when we put iodine on the solution, or you can say on your potato which contains lots of starch, the iodine yellow brown color converts into black coloration. So it proves that yes, iodine is present. Now we are going to perform the third test, and that third test is going to be for the lipids. We all know that the lipids are unable to be dissolved in water, so that's why we dissolve the liquid lipid, or you can say the solid fat, into alcohol first. We shake it and then we try to form fumes on the top. If there is a fumation formed on the top of your water, like represented by this white color, this fumes will represent yes, fat is present in alcohol and it has been separated, and now it is forming this fumation layer. The last test is to detect the presence of proteins. Protein is present in egg white, we all know that. So for egg white, we take equal amount of egg white and biuret solution. It is also of blue color. Do remind that, lekin this blue color is going to be turned into violet color as soon as it reacts with protein. So the violet color presentation indicates that yes, protein is present in a very high amount. Thank you for watching. Please comment if you have any questions.